Hello everyone. Today we will learn about how to download sequencing data from NCBI as well as from other repositories. To download the sequencing data, we will use NCBI's SRA toolkit. SRA toolkit has many functions such as prefetch and faster queue dump is used to download the data from the database, and VDV dump is used to extract metadata that are associated with the data. Right now, we are on the homepage of SRA database. SRA is Sequence Read Archive, and this is the largest publicly available repository for high throughput sequencing data. It also integrates data from different sources such as DDBJ, EBI. Any sample that has been submitted to SRA database has an associated SRA ID. We can search this database using the SRA ID, as well as we can also write the type of data we are looking for, such as RNA-seq data, human. This is showing us the data. Now I will show an example how we can download the data from SRA database using SRA toolkit. To start, we need to install the SRA tools from Anaconda. We will copy this line. Now we will paste this line on terminal. This is telling that this package is already installed because I have already installed this on my system, and conda env list is it is installed on my bioinformatics environment. If it was not there, it would lead to finish the installation. In this directory there is a text file that contains the SRIDs. There are two SRIDs. We will download those files using SRA toolkit. Let's try to download the SRA file using prefetch. It is downloading the SRA file. It could take some time depending on the size of the file. The download is successful. Let's take a look. There is a new directory created with this SRA ID name, and there is a file that is ending with .sra. This is a compressed binary file. From this file, we will extract the raw FASTQ files. We will use FASTQ dump command to get the raw FASTQ files. So it has extracted the raw FASTQ files. Let's take a look. Yes, there are two files. This was a paired in library. For that reason, there are two files for forward and reverse read. So this is how we can use prefetch and faster queue dump command to download raw FASTQ files from SRA database using SRA toolkit. In addition, I will show another example how we can download a batch of files from a list of IDs. In this file, we have two SRA IDs. Let's write a while loop to download both of these files together. Yes, this is printing all the lines in this file, and now we can use prefetch. For each ID, this will download all the lines that is in this file. In this case, we have two lines with two IDs, and this is downloading the .sra files. If you have like hundreds of SRA IDs, you can put in this file, and using this while command, we can download all the SRA files. Yes, both these directory have been created based on the SRA ID that we have provided in the text file. Now let's take a look inside those directories. for both the directories whether there is an SRA file downloaded or not. Yes, inside both directories there are SRA files. Let's now use FastIQ dump to extract the FastQ files from both SRA files. File in this command is reading all the SRA files. Now we can use FastIQ dump. This will lead to dumping all the FastQ files from SRA files. The FastQ files have been generated in this current directory. It is important to note that for this SRA ID, there are two FastQ files because this was a paired end library. And for the other ID, there is only one file because it is a single end library. There are other options available in the FastIQ dump program. We can specify the output path. We can also specify how those files or the data will be split. And there are many other options available on the FastIQ dump. I would recommend to check those options. If we type with -h, it will print all the options available. Also, I want to note that it is important to learn basic bash commands, for example, while loop, for loop, and how to access directories. I have made a separate course on basic bash commands. Please take a look into those videos. Thank you for taking the time to watch this video. See you in the next one.